Hereby I open this academic ceremony in which Ms. Jian-Hua Cao will defend the academic thesis entitled Local Metabolism in Preclinical Disease Models Studied with Mass Spectrometry Imaging. May I invite you, dear candidate, to present a summary of your studies and the conclusions of your thesis in the next 15 minutes.
Thank you, Pro-Rector. Dear Pro-Rector, dear committee members, dear family, friends, and audience, it's my great pleasure to present to you my PhD work entitled Local Metabolism in Preclinical Disease Models Studied with Mass Spectrometry Imaging. As we all know, our organs are working together to keep us healthy. And there are diseases that affect specific organs in our body. Take atherosclerosis as an example. It's a leading cause of death and disability all over the world. It's also a lipid-driven inflammatory disease characterized by plaque formation in the arterial wall. Plaque formation involves very complex cellular activities in different layers of artery, especially intima and medial layers. In short, plaque formation starts with endothelial cells dysfunction and accumulation of circulating lipoproteins and inflammatory cells in the intima. Moreover, smooth muscle cells from medial layer can also migrate into intima, leading to a larger and more complex plaque. Plaque can continue to grow, and it could rupture, cause myocardial infarction or stroke. In summary, plaque formation involves a very complex cellular network. Moreover, as we all know, within a cell, there is thousands of metabolic reactions are taking place, in which a wide range of metabolites are involved. Therefore, we need a high-throughput technique which can capture both cellular and molecular complexities to a better understanding such complex disease. Mass spectrometry imaging is an imaging technique that makes it possible to in situ detect thousands of molecules simultaneously while preserving the tissue's morphological integrity. Therefore, it captures both cellular and molecular complexities. A mass spectrometry imaging experiment is performed in an XY grid-wise manner. A mass spectrum of every spot is acquired. This set of mass spectra allows for image reconstruction by showing the abundance of each MC value as a function of each mass spectrum's coordination across the raster tissue sections. This results any MD species can be spatially mapped. The exact same tissue section can thereafter be stained histologically, such as hemotoxylin and eosin staining, and co-registered with MSI data. Due to many advantages of this imaging technique, we applied it to study metabolomic alterations in atherosclerosis in one widely used mouse model. Spatial metabolomics data were obtained from 11 control mice and 11 pathological mice. The results showed metabolic alterations is not just happening in different layers of artery, but also in the surrounding heart tissues. Moreover, most of these significant molecules are more abundant in the pathological condition. This is a visualization of two selected molecules. You can see the molecule which represents in pink color is more localized in the heart tissue, and it's more abundant in the control group, while the other molecule, which represent in green color, is more localized in the plaque region in the pathological group. Further molecular assignments highlight the roles of lipids in atherosclerosis, and metabolomic alterations is not just happening in the artery, but also in the surrounding heart tissues. In the next step, we would like to investigate our translational potential of our preclinical results from tissue to a clinical diagnostic setting in human plasma. This is currently ongoing with our collaborator in Spain. As I mentioned earlier, LDLR mice were used for the study I just introduced. However, ApoE is also one of the most widely used mouse models in atherosclerosis. Since these two mouse models have their both advantages and limitations, identifying common molecular features harbours a high potential to a better understanding such complex disease. Therefore, in the following study, we aimed to identify plaque-specific lipids in both mouse models using two-dimensional and three-dimensional mass spectrometry imaging. Two-dimensional MSI data were obtained from 15 LDLR mice and 4 ApoE mice in two different ionization modes. This results in a total of 25 MZ species were found significantly and specifically co-localized in the plaque in both mouse models. Of these, 11 lipids were positively identified. This is a realization of one selected plaque-specific lipid. What is more interesting is that we found this lipid is more localized in the necrotic core inside of plaque. This observation is consistent in both mouse models. In the next step, we would like to investigate the volumetric distribution of these plaque-specific lipids using three-dimensional mass spectrometry imaging in one ApoE mouse. This is a 3D reconstruction of one selected plaque-specific lipid. You can clearly see this lipid is present in the plaque from beginning to the end along the Z direction of the aortic root. This is another direction. We look at the 3D data. This is a 3D longitudinal view of two selected plaque-specific lipids. You can clearly see they are present in the plaque from beginning to the end along the direction of blood flow direction. Moreover, we performed the correlation analysis section by section of a 3D data set, which clearly say the spatial correlation coefficient remains stable along the Z direction of the plaque and is independent of blood flow direction, which further confirmed the universal presence of these lipids in the plaque. In summary, so far we have been investigating local metabolism in atherosclerosis. However, as we all know, metabolism is not just local, but also dynamic, which means it can change over time. Now I would like to direct your attention to another pathological condition called wound healing. In order to investigate both local and dynamic metabolism, we applied mass spectrometry imaging to wound healing process involving several time points. Similar to atherosclerosis, wound healing is also very complex and dynamic, involving many cells and molecules. In this study, we aimed to apply mass spectrometry imaging to investigate lipid alterations during the abdominal fascial wound healing process using mass spectrometry imaging, and later correlated with the histologic alterations during the wound healing process. This results in a total of 5 lipids were found significantly involved in the wound healing process. This is a realization of 2 selected of this lipid. You can see they are more abundant in the inflammatory region. In short summary, So far, we have been focused on local and dynamic metabolism using mass spectrometry imaging in 2 different pathological conditions. You may start wondering, can we also address systematic metabolism in addition to local and dynamic metabolism? If the answer is yes, how can we do it? Well, this brings me to my next research chapter entitled Mass Spectrometry Imaging of Isotope-Labeled Phenylalanine and Tyrosine in the Context of Cancer Tissue in Mouse Models. This approach combined mass spectrometry imaging with isotope labeling of targeted molecule, which makes it possible to investigate molecular synthesis and turnover in a spatially resolved manner. And this approach has been established earlier in our lab to track hepatocellular incorporation of isotopes phenylalanine and tyrosine in liver. In our study, we applied our previous established approach to investigate isotope-labeled phenylalanine and tyrosine in cancer from same animals and involving same time points. The results showed most of unlabeled amino acids, together with isotope-labeled phenylalanine and tyrosine, showed similar spatial distribution, with a higher abundance in the viable tumor compared to non-viable tumor. If we take a look at isotope-labeled phenylalanine and tyrosine over time, you can see highest enrichment of isotope-labeled phenylalanine was detected at 10 minutes and decreasing over time, while the overall enrichment of isotope-labeled tyrosine was substantially lower, and it has a delayed trend with its peak at 30 minutes. This similar trend has also been observed in both viable tumor and non-viable tumor separately. Later, LSC MS data from homogenized tumor sections also confirmed the similar results of MS data. The additional plasma experiment from same animals showed both isotope-labeled phenylalanine and tyrosine reached their highest level at 10 minutes and decreasing over time. This, together with the observation that highest enrichment of isotope-labeled tyrosine in tumor was delayed, indicates that isotope-labeled tyrosine was later transported to tumor after a phenylalanine to tyrosine conversion process, which is assumed predominantly occur in the liver. In summary, this study investigates isotope-labeled phenylalanine kinetics in cancer, and it also reflects amino acids inter-organ metabolism. Taken together, all of these efforts give evidence of usefulness of mass spectrometry imaging applied in animals to investigate local, dynamic, and systematic metabolism in different pathological conditions. Well, most of these studies are technically based on non-targeted mass spectrometry. The first atherosclerosis study showed such approach can also lead to findings that can be later exploited by targeted mass spectrometry. Finally, some of the approach used in this thesis can also serve as blueprint to study other diseases. In the end, I would like to say thank you to the whole M4I family, especially my supervisors, Ron and Benjamin. I also want to say thank you to my collaborator from Maastricht UMC, especially Eric and Judith from Pathology Department, Nico Steven from Surgery Department. I also would like to say thank you to my collaborator from Spain, especially Gloria and Malada. Thank you all for your great attention. Now I want to give the word back to the prorector.
Thank you very much, Ms. Cao, for your excellent presentation. Very detailed and very scientifically content. We're going to start now the opposition, and for the audience, we have 6 members in the opposition, and we're going to start with Professor Honing. Professor Honing was the chairman of your assessment committee, and he's affiliated within the Department of Analytics for Systems Imaging at our university, and I'd like to give him the first word.
Thank you, Mr. Prorector. Dear candidate, first of all, congratulations with this investigation into the boundaries of application of mass spectrometry images, especially in metabolic systems. And I particularly liked also this thesis because you entered a nice puzzle for us. So I was reading at page 48, and I thought, well, that's a nice puzzle. I'm going to try to find it out. So if you look to this table, I think it's table 2.1, and there is a blind spot, and I tried to find out in this puzzle what should be entered there, but I could not, because I tried to think about LPC 18.2. But can you help me out? What should be in that blind row of information? I could not find it.
Highly esteemed opponent, thank you for your kind word, and thank you for your question. Would you mind repeat the question again?
Yeah, so on page 48 in your thesis, there's a nice table. And I thought first it was a puzzle for me, but maybe I'm wrong. And on the 4th row, there's no data. So can you help me out? I tried to fill it in, but I could not.
First row?
The 4th row.
Oh, yes. Highly esteemed opponent, thank you for your question. So this is identification of our plaque-specific lipid. We got at least like 25 MC species. Then we try to do MS1 and MS2 using high spatial, high mass resolution mass spectrometer to try to identify those lipids. However, this, for example, this molecule, which we could not find in any database, or we could not identify what's the identification of this molecule. So that's why we put it black, because no one knows what's that. So I put there.
So that's anyway a good starting point for new research in trying to find out what this molecule is.
Yeah, it could inspire other researchers, or maybe from our lab, or from other.
That's very good. It's always good that your thesis is inspiring other researchers for new research.
Yeah, so that's the point. Thank you.
Very well done.
Yes.
I would like to go back to page 85, and I first will ask you, and you mentioned a little bit in your introduction, the word dynamics, because in metabolism you use the word dynamics. I would use other words, but what do you think, what do you define as dynamics?
Highly esteemed opponent, thank you for your question. So dynamic, let's say in my thesis, we try to, actually in every chapter it shows dynamic. For example, in first study, we use 2 mouse models. We study, they have different diet in end, and they add up with plaque in their artery. We try to find what kind of lipid accumulated there. In chapter 3, we even go a step further. We try to find one is control group, one is high fat group. We see over time what kind of molecular changes at their different layers of artery, even and heart tissues.
Yeah. So you agree with me that dynamics here is the constant change of concentrations of certain metabolites in your dynamics or in your system?
Yes,
yeah?
It's a constant change because of external elements or environmental, it's metabolized very highly dynamic in that sense.
I completely agree anyway, so that's not the problem here. But on page 85, in the second alinea, in the 3rd sentence or line, you start to say that genomics data is not able to reflect on the dynamics of metabolic processes. And then I'm a little bit puzzled, because metabolic processes speak about the change of the concentration of metabolites. And now I would ask you, what is the reason why this change happens? Why do these reactions happen? On basis of what are these reactions occurring?
I think for genomics, at least in cancer, 5% to 10% cases can be highly related to the gene changes, gene mutation. However, like 90% related to the environmental or diet issues. So we refer to here like only small amount of patient can be traced back to the genetic changes. However, most of the cases, they are related to the environment or diet. So
but there I would really interfere a little bit, because the genome defines the subscription of proteins, and I think the catalysis of all the metabolic reactions taking place in your body are on basis of enzymes and so protein. So any change in protein structure dependent on genomic changes is of course influencing the stability or the dynamics of metabolic processes. Would you agree with that?
I would argue that some metabolites can also react with proteins in that it could change the gene expression. So no matter genomics, transcriptomics, proteomics, or metabolomics, they are working together to make this disease progression.
Absolutely. So
yes.
I completely
agree. I think everything should work together.
Voilà.
I would not say genomics can make this disease happen. Everything's working together.
Voilà.
Yes.
But then the statement is a little bit harsh, but you agree that it's a little bit more complex. That is the thing. I would like to go to page 88. You have a nice figure, 5.2, on, and especially you would like to prove the distribution of this stable isotope labeled phenylalanine and tyrosine. And you use also the word some, for some amino acids there's a difference, and for some no, and you say yeah, most, but still there's 3 amino acids like asparagine and glutamine which do not play a role here anyway. But do you understand why some amino acids are significantly in the tumor cells or tumor part and some not? And linked to that, there's also a lot of amino acids, particularly in this necrosis field, also the tyrosine, but not the labeled tyrosine. So I think there's much more research to be done in these very complex pictures.
Yes, I totally agree. Actually, this figure is really perfectly show why we need to study disease in a spatially resolved manner. We found some amino acids. You saw most people think, oh, viable tumor, all the metabolites should be there. But in that, we found even some amino acids more localized in the necrotic core. Actually, so many things happen because during this necrotic core build up this necrotic core, there is so many immune cells involved in the whole process. So there is so many things you need to do to figure out what's going on there.
Okay. No, thank you, Ms. Broder.
Okay, thank you very much. Then the opposition will be continued, and we go to Professor Shiri Svetlov. She's affiliated to the Department of Hepatic Information and Metabolic Health at our university, and she was also a member of your assessment committee.
So, dear candidate, first I would like to congratulate you for your very clear presentation, which also very nicely demonstrated your passion, and also to extend my collaborations to your promoting team for your excellent book. I personally read it with lots of interest. I think you very nicely demonstrated how we can use MSI to better understand the metabolic diseases and possible applications of that. So congratulations for that. So my first question is about chapter 2. In this chapter, you compare the LDLR knockouts to the ApoE knockout mice, and you looked in the common lipids within the plaques. Also in your presentation, you showed very nicely the differences between the two models, and especially a big difference is that the ApoE knockouts are on chow diet, while the LDLR knockouts are receiving the high-fat diet. Taking into account these differences, did you expect to find such big similarities in the lipid composition in the plaque?
Highly esteemed opponent.
Maybe you can move a little bit away from the microphone.
Sorry.
No, not that far, but not like this.
Okay.
A little bit.
Yes.
This guy.
Thank you. Yes, no problem. Yes. Highly esteemed opponent, thank you for your kind word and thank you for your question. To be honest, because these two mouse models are two or most widely used animal model in atherosclerosis, we look up many literatures, and actually the description of the differences of these mouse models is not very clear, and there is really few studies to try to investigate the both similarity or differences in these two mouse models. If we trace back to the mechanism of how this atherosclerosis happening, is because the endothelial cell dysfunction and accumulation of lipoprotein and also inflammatory cells in the intima. Later, as most muscle cells also migrate into there, the whole process, the similar cells involved there, even no matter LDL knockout or ApoE knockout, they all play a role in the whole atherosclerosis development and progression. So we are not very surprised there is similarities, but we also observe some differences. For example, ApoE, we found more single lipids there, but in general the similarity was kind of high, and also in the we checked also the HE staining. We found they are more or less more advanced plaque. So we are not really surprised.
Yeah, but I mean, what is then the role of the diet? Because I mean, the lipids in the plaques, are they coming from the diet? Or if that would be the case, then why would you find the same lipids in the two models?
So one reason is, as I mentioned earlier, not many studies to try to investigate both mouse models. We are just, first we want to send an example. We can use mass spectrometry imaging, no matter 2-dimensional, 3-dimensional, to investigate this disease. Secondly, we also want to find out even different knockout mechanism can also lead to similar lipid patterns, even some lipid which really consistent.
So it could be, for instance, related to the fact that both are hyperlipidemic models, maybe.
Yes, it could be, and also we want to also later translate this result to human, and we also look at the data from human plaque. Actually, most of these lipids also found in the plasma in human patients. Okay. So I think the results is quite convincing.
Okay, and is there something you can learn from, because in chapter 3 actually you have the LDLR knockout mice who are fed on either chow diet or on a high fat diet. So is there some kind of general conclusions that you can say about the diets when you combine these two chapters together, like something that comes from the combination?
Dear highly esteemed opponent, thank you for your question. So actually, the chapter 3 is inspired by chapter 2. Chapter 2, we try to find mainly focus on plaque, but chapter 3, we just try to find what's going on, what's happening in different layers of artery and also in the surrounding heart tissues. Most people they don't really look at the environment, like surrounding heart tissue, but it's very intriguing us. We also found really a lot of lipid alterations there, and also we have to remind people here, like in the chapter 3 we use the early stage, a bit earlier compared to chapter 2. So that's the reason we want to push a bit further to try to translate our results to clinic.
So could it be indeed in your discussion in chapter 3, you mentioned that the main differences in the lipid composition that you found was that because it's advanced versus more early plaques, so the differences in the duration of the diet. Could there be other explanations for that? What other differences that you have between the two setups?
Another difference is actually we in the end we also translate those results to human plasma. We actually combine these two studies because we found some lipids. For example, in chapter 3, we found some plaque a very early stage, some plaque a very advanced stage. Later we also try to compare what's going on there, and we found these lipids also in the human plasma. So it's I think it stems forward to translational potential. The chapter 2 is more mechanism.
Okay.
Yes.
But could it be also the composition of the diet that is slightly different? Did you look into that? Because in chapter 2 you have 0.15% cholesterol, and in
Yes, could be. Yes, could be.
okay. And I don't know if I have time for another quick question. No. Okay. Thank you.
Thank you, I'm sorry. We have to continue. And we continue with Professor Andren. He was also a member of your assessment committee, and he's affiliated professor of mass spectrometry imaging at Uppsala University in Sweden. And I'd like to thank him that he joins us this morning. Thank you.
Thank you, Dr. Prorector. And thank you very much for a very nice presentation and a thesis that was enjoyable to read. I have a question, or a couple of questions regarding your propositions, which also I think was very to the point regarding this thesis and the research you have done. So I have a question regarding proposition 1. So maybe we can have that.
Yes. Okay, yes.
Shall I read?
She's going to read.
Oh, sorry. Yes.
Proposition 1. Animal models are a fundamental tool in preclinical studies for studying human disease, this thesis.
Right. So I would like to know, and I would like you to comment on, if you know, how many promising preclinical studies that are successfully translated into clinical applications. Do you have any idea? Is it 50%, 100%, 1%?
Dear highly esteemed opponent, thank you for your kind word and thank you for your question. I think from animal model to clinical, the percentage is very, very low. I'm not sure, maybe it's even less than 1%. Yes.
Well, that's there are many reports out there, of course, but I think around 5 to 10% is the number I've seen, and I ran into an interesting paper actually that I have to read the title of the paper. Lost in Translation, The Valley of Death Across the Preclinical and Clinical Divide. So there are some issues there, you know, translating preclinical data to clinical data. And in chapter 3, you have studied, you have used both animal data and clinical data. For example, in the animal study, you found that N-steroyl glutamine had a prominent role in initiation of atherosclerosis. But you also measured plasma with LCMS. And there you also found some interesting molecules such as carnitine and 3-methylamine N-oxide, TMAO. So my question to you is, did you actually also look into the plasma data and try to use that data in the mass spec imaging data to verify your clinical data to the preclinical data, so going the other way around?
A highly esteemed opponent, thank you for your question. Indeed, we look at the data, both MSI and also plasma. So in this chapter, we did not end our additional experiment which we performed based on the MSI data, because as I mentioned, in the ALTA, there is different region which grew plaques. Actually, some region have more advanced plaque, some region are very early plaque. We asked our pathologists to try to annotate these both regions. In the end, we found actually 2 lipids which are really positively identified. One is LPA, one is LPC. They are very highly and significantly abundant in the advanced plaque. Later on, we already performed the experiment in human plasma using LCMS. We found these 2 lipids also highly abundant in the patient with high risk of cardiovascular disease. So in this case, we found these 2 lipids, at least they are both show a higher tendency in the pathological condition, and also we look at the data from other publications. These two lipids, and also many people try to study them in the clinical setting in plasma or serum. They found these lipids show a poor prognosis of this disease. So I can say no matter in tissue, which is more in the direct local of where the disease is happening, and in plasma level, which we look at in systematic metabolism, these two lipids really play a role in this disease. I will be very happy to see these lipids really go into the clinic. Yes, thank you so much.
Thank you. Because, for example, you showed that carnitine was changed in the plasma between the two groups, and carnitine is a molecule that you can also image quite easily. Yeah.
Yes, I think so, because it's one type of amino acid. We could use derivatization to, or just no derivatives. I believe you can use multi-FT-ICR, high mass resolution instrument to look at this molecule. Yes, I think it will be very interesting to look at them.
Right. I also have a very technical question regarding that study in Chapter 3. You report that you detect 2,901 mass charge ratio values. How is that calculated?
Highly esteemed opponent, thank you for your question. So in the workflow we used at least in my different projects, we measure everything using the same instrument. Then later on we did the correlation with the staining. Then we import everything in the Scales. So Scales Lab is a software I always use for the later on data analysis. We export the overview average spectrum, then we imported it in the MMass software, and we did a peak picking there, like isotope removing, or we just choose some signal to noise threshold. In that, we got a more positive list, which show a high abundance on tissue. Then we use this list. Later on, we do the different data analysis. That's how those numbers come out, yes.
But it wasn't 2,901 in each individual experiment.
I believe there is more than that. It's just in the end, we combine whole data set together. We want investigate, more focus on this more abundant molecules.
Right. Next question is also about Chapter 2, and it also relates to your Proposition 1. You measure lipids in the plaques, right, in Chapter 2, and you also measure the lipids in human plaques, but you also state that the human plaques are more complex than the mouse ones. So is it really necessary then to do the animal study, since you already have the data which is more complex than the animal study?
Highly esteemed opponent, thank you for your question. To be honest, in my thesis, in the end, I only got one plaque from human sample. So if you look at my chapter in discuss chapter 7, I believe, we did MSI, all this one human plaque. We showed those plaque-specific lipids from mice. We also observed in the human samples, and it showed very heterogeneous distribution. Regarding to necessary, I think it's very necessary because in Chapter 2, we use 2-dimensional and 3-dimensional mass spectrometry imaging. The advantage of using 3-dimensional mass spectrometry imaging, we cut through the whole plaque along the z direction, the L-tic route. So, which means the data is, those lipids very robust. You can really maybe use this data to the clinic. And it also reduces the sample bias because it's really an issue in clinic. If the tissue you're not get the right place, you might get a bad conclusion and it will not get a good treatment. I believe my research on chapter 2 is very convincing and is very valuable to the, no matter preclinical research or the clinical research, it's very valuable. Thank you. Yes.
Thank you.
Sorry, I interrupt. The time is over, so we have to proceed to the next opponent, which will be Professor Jonkers. She was also a member of your assessment committee, and she's affiliated with the Department of Intestinal Health at our university. I'd like to give her the word.
Dear candidate, I would like to congratulate you and your team. It's really a fantastic thesis. It was really a pleasure reading. I think it was also very well written. I would like to stress that as well. I was really impressed by, I think you're a good networker because I think you combine a lot of techniques. You work with a lot of disciplines together because you had a lot of co-authors from different groups. And I also was impressed by your social network with a lot of international contacts and having worked together with a lot of colleagues. So, and your work interests me a lot. I think it is a big step forward, but it also triggers me how we can use it for the human research and can learn from that and follow up a little bit on position 1 and the discussion you had. And I would like to go to chapter 4 with you, where you did study wound healing. And maybe first for myself and my curiosity, a small methodological question to see if I understand it correctly, because you actually looked to the overlap between the histological annotation and the MSI profile, and then you did a Pearson correlation and had a threshold of 0.4, and I was just curious, is that really good enough, a 0.4, to really make this, or do I understand that wrong?
Highly esteemed opponent, thank you for your kind word and thank you for your question. Actually, for we perform data analysis called correlation analysis. You can put the threshold whatever you want. However, in most of cases, we try to put the threshold a bit higher. Of course, if you put 1, which means it absolutely there is absolutely positive, absolutely 100%. However, in most of cases, we need to take into account our data and also try to make the data analysis and also later interpretation make sense. So 0.4 is in our field is quite significant, and its correlation analysis is based on mathematical calculation of two images. One image is the intensity image of one selected mass. Another is a binary image, which shows where the target tissue is. Okay. So, and it's also taking into account the size of the region we are interested in. So in the end, 0.4, we also the similar analysis we performed also in the chapter 2. You can see the results is quite convincing, and we see.
Okay. You absolutely convinced me. I'm very happy with that answer. Then I would like to go to the model you used, because what I noted that you would like to study wound healing and also impaired wound healing, because you indicated that it's really a clinical problem. And then you have an animal model from animals which are all healthy. You even indicated that they are not using medication, which patients often do. And then you did not find complications in 34 out of the 35 rats. So I was wondering, is this a good model for wound healing, or how could you improve that? And for impaired wound healing especially.
Highly esteemed opponent, thank you for your question. Regarding to these animal models, actually is as I mentioned earlier, it's a collaboration with people from surgery department, and those animals also used for other research, to be honest. And I believe their group also performed some genomics analysis. we found a similarity with the human wound healing process. In this approach, we are not looking at the chronic wound healing process, and we are more try to find the mechanism of normal wound healing process, which that's why we use healthy animal models. Because most of the healing process involving the same process, 4 stages involving complex cells and molecules, and we try to find during the normal, without any complication process, how this molecular mechanism or at the cellular level, how it changes. So we try to find this mechanism, and later on it could help for the people who want to study chronic wound healing process, because they are quite similar. It's just it could delay one stage to another stage, but the whole process is quite similar. That's the main purpose of this research.
Okay, I understand you focusing on the normal wound healing, because it could also be interesting to compare those with impaired wound healing and normal wound healing. Because then at the end of your abstract especially, you concluded that you found this glycophospholipids and gangliosides to be involved in the normal wound healing, and they could be used as potential markers. Can you convince me whether you have sufficient evidence that they already can be used as potential markers for wound healing in the clinic? Do you have enough data for that?
Highly esteemed opponent, thank you for your question. Positively, yes, I could say yes, because in clinics, for example, phosphocholine lipids already applied in clinics, also in preclinical models. They put all the wound part, and they see the healing process a bit fast, and patient get a good recovery. And so we found a similar lipids, not exact same, but similar lipids. We also look at other data from other people. They study different cell mechanisms, and they also try to study if those lipids could help during this healing process, and they are very positively concluded. So I believe if we try to push a step further, it could apply in clinics. I'm not here. We would not assume that it will, but I think give the similarity of these lipids to other researchers which already used in clinic, I believe it could have a positive outcome for the clinic use.
Okay.
Yes.
Do we have time for a short other question?
If it's short, yes. If it's long, no. Short.
Then I'll keep it very short, because also intrigued by chapter 4, chapter 5, where you studied the labeled phenylalanine, and you can answer that then also very short. Is this also in a technique we could apply in humans itself?
I believe yes.
Okay, thank you. I'm quite happy with that answer. Thank you very much.
That's indeed a short question and a very short answer. Thank you very much. We will continue with Professor van Soest. He was also a member of your assessment committee. First of all, I would like to welcome him, that he came all the way from Erasmus Rotterdam to join this defense. And he's affiliated with the Department of Invasive Imaging, in particular cardiovascular diseases. And I'd like to give him the word.
Thank you, Mr. Prorector. Dear candidate, I would first like to congratulate you with the completion of this thesis, which I read with great interest. And I would also like to include your promotion team with that fantastic result. I would once again like to turn the discussion to chapter 2 and 3. The first issue that I would like to discuss with you is that, so you find a range of lipids that indicate contrast between your disease group and control group, where the control groups are actually different between the two chapters. But what strikes me is that the list of lipids that you end up with is quite different from what we see in human pathology that have been published. So the human studies see, they do see sphingomyelins, like you also report, but they also see a lot of diacylglycerols, triacylglycerols, cholesterol esters, phosphatidylcholines. Can you share your thoughts on what might cause that difference?
Highly esteemed opponent, thank you for your kind word and thank you for your question. Indeed, we see some differences, and the reason can be many. The first, overall, we are using animal models. As I mentioned, animal model is really different from human, no matter even in genomics or anatomy, even molecular cellular level, they are quite different. They are similar, but there is differences. That's, I think, one big reason. Another reason is human is very more complex condition, because animals they have really controlled diet, and they are really in the stereo environment. For a human, we are really in a very different environment. So many elements can influence liquid content.
You say it's a difference in species and environment. But if I go to your well, admittedly it's an n=1 example in chapter 7, you indicate that you see the same lipids returning in the plaque, and so the wide variety of lysolipids is not so clearly identified in literature so far. So I would like to. So could there be experimental differences also playing a role?
I believe yes, and we also look up other researchers. For example, actually, your group did a lot of study about atherosclerosis. We use different matrix application and also a sample difference, and also later data analysis. Also, identification process is all different. I think they kind of play a role there. Yes.
Okay. Good. Well, that actually is a nice preface for my next question. So thank you for that answer. So between chapter 2 and chapter 3, you actually take quite different experimental approaches to studying a very similar problem. Can you, so in chapter 2 you use a RAPI and Noraman as a matrix, whereas in chapter 3 it's a Solarix and 9AA. Can you comment on why you made that change, and whether there's any impact on the data that comes out? How should we factor that into the interpretation?
Highly esteemed opponent, thank you for your question. Indeed, we use two different instruments in two different chapters. In chapter 2, we use RapidFlex for the two-dimensional mass spectrometry imaging experiment, because the instrument is quite fast. And later on, we use Orbitrap to do the identification. It's quite robust because it's really high mass resolution. In chapter 3, we go a step further. We use FT-ICR, which really, I think is the highest mass resolution.
Why did you make that change?
The reason is because we know identification of metabolomics is a huge challenge in the whole mass spectrometry imaging community. Even in the bulk metabolomics community is also very high challenging. We use this high mass resolution instrument to help us later on if we try to identify as small as possible. So if we got a really high mass resolution and MS1 level, it could help us to identify more molecules.
Without having to add an additional experiment with
yes, because
MSMS on
because
extracts.
We also have another issues, because it's a collaboration again. We got the animals from pathology department. They really like OCT, but we hate OCT, but they use OCT. We also we did additional similar to chapter 5. We use derivatization, use THS to see to increase the ionization efficiency of amino acids. We also try to do that, but OCT it can melt really a little bit high temperature. It can melt and it's go everywhere and really influence our results. So it's a bit sad, but it happens. So we can do nothing about it.
Those are not easy experiments. Yes. I can concur. So I have one final question regarding your results on the contrast that you see in cardiomyocyte metabolism between LDL receptor mice and between the normal diet and the high-fat diet. So I'm curious about the mechanism, because we have a mouse which has an LDL receptor defect, yet you say that this the diet then causes a change in the cardiomyocyte metabolism. You hint on ischemia maybe playing a role? Do you have independent evidence of ischemia actually occurring in these mice?
Highly esteemed opponent, thank you for the question. And in our research, we did not perform additional experiment to confirm its ischemia condition. We did some research according to other literatures. It could we have assumption maybe the heart is also in an ischemia condition. It will influence the lipid consumer production. So it's an assumption, but we are not say.
So what reason do you think do you have to not think it's simply a diet effect?
It could be. So because it's really to control its normal diet, pathological condition is really high-fat diet. Is for sure the diet play a really key role there. But we also try to find because it's mainly in most cases we think it should be more changes in the artery tissue, but it was very convincing, striking us it's also happening in the heart tissue because it's not directly at the site where the disease is happening. So we try to think and what's going on there, and
so
yes.
We're still in the hypothesis generating
yeah,
phase.
It inspire other researchers to do some research on that, yes.
I assume my time is up, right? Okay,
I'm
thank you very much.
Exactly. Thank you very much. So the opposition will be closed by our last opponent, which will be Dr. van der Vorst. He is both affiliated with the RWTH Klinicum Aachen department and the Department of Pathology here at our university, and I'd like to give him the final words for this last opposition.
Yes. So thank you very much. First of all, many congratulations with this very nice thesis. I also want to extend this to the promotion team. So I'm a pathologist, immunologist. My level of expertise in MSI was very limited. So I find your introduction very educational, very well written. So thank you for that. Since my background is also in the biology, I also want to switch a bit more towards this direction. And in chapter 2, you focus on this LPI 18-0, and then you say that this lipid it's mainly located in the necrotic core in these mouse plaques. Then in the discussion, you wrote a sentence that triggered me, where you state that the specific role of this LPI in necrosis still has to be explored with other techniques. Can you speculate? What can the role of this LPI in necrosis be, and how would you investigate this?
Highly esteemed opponent, thank you for your kind word and thank you for your question. Indeed, it was also intriguing us, this lipid we found really is more really abundant in the necrotic core. And if we check the data from other biology experiments, we see this lipid is play a role in the different stages of atherosclerosis. For example, endothelial dysfunction, inflammatory process, and oxidized low-density lipoprotein, and also the plaque rupture. And it also, this lipid, I believe, is also a prognostic marker for the myocardial infarction. We try to think how can we do that? Then, yes, I have some ideas. So, for example, we could do some cell culture experiment. For example, we incubate this lipid with inflammatory cell. Let's see how it could influence the necrosis process. I believe there is marker in the biology department. There is a marker which could investigate if this lipid could promote the apoptosis or necrotic process. Another experiment we could also do is we go step further. We do it on animals. We could add this isotope level, the lyso-PI lipids, and let's check at different time point how this necrotic will grow and different cellular level and molecular level. Let's see how it involved in the whole process. And another idea would be we could also, I believe in the pathology department, there is a technique which you can apply such lipid in prevascularly I think use a gel. I don't clearly remember which gel it is, but you can put all the vascular, then you can see if this LPI challenge the vascular process. Let's say if this lipid necrotic core is going up or is going stable or not stable, because necrotic core is really like risk for the unstable plaque. It really play a role in the fatal event. And another idea, I would say multi-omics, because it's really a hot topic in the all field. We can combine genomics, transcriptomics, lipidomics, metabolomics, and try to say this LPI, I believe it's a specific bite to the G protein receptors. I think it's called the GPR55. It's really play a role not just in the cardiomyocyte and also endothelial cells and smooth muscle cells. It's, we could use genomics to find the genes which regulates this lipid. Let's say if this gene is up-regulated or not, it could explain why it's play a role in the necrotic process.
I totally agree that will be very interesting experience to do. And but when also if I then looked in the chapter to figure 2.5, where you have to show this longitudinal 3D view, you also show nicely the topography of the lesion. And then if I then especially look at these shoulder regions of the plaques or the edges, you also still see a very high level of this lipid. But in these shoulder regions, they're very active, but they're not so much necrotic. Why do you think that this lipid is then also present in these shoulder regions of these plaques?
A highly esteemed opponent, to be honest, we look at the section by section and the morphological condition. We also found those necrotic core there, and also because this lipid is, as I said, is really distributed from beginning to the end. Yes, so no matter it's shoulder or end, it's always there. So I would say it's really robust lipid in this field. Yes, thank you so much.
Mr. Candidate, if you want to add an additional point, you're free, but if you said you answer it, then we just close it. It's up to you.
So I want to add that we end this topography just show we cut really through the whole plaque, and we even did additional 3D and also data analysis to show this lipid really present from beginning to the end, and it's really robust, and no matter you look at which any direction, it's always there. So that's my answer.
Thanks a lot.
Thank you. Thank you very much. Ms. Cao, the time appointed for defending your thesis has now passed. The degree committee will now withdraw to discuss not so much the quality of your thesis, because that has been approved, but the way you have defended your thesis this morning. I request that you and your company await the results of our deliberations and our return to this room. Thank you very much.
Thank you.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes. In the meantime, let's take a closer look at Maastricht University's renowned teaching method, problem-based learning. Once the prorector reconvenes the session, we'll tune in and continue the live stream.
Problem-based learning, or PBL. What does that mean exactly? 3 of my fellow students and I will show you around. Every week, we analyze a different case or issue together. We discuss the case, and everyone can contribute different perspectives to the group discussion. If we get stuck, our tutor helps us out and suggests what we could do next. I prefer going to the library to prepare. Here, I can focus, and I have quick access to books or journals that help me understand the case. Today, I can also train my stitching skills in the Skills Lab. where you can immediately put into practice what you've learned. After a day like this, I like going to the gym to clear my head and get ready for the next day.
At UM, you meet people from all around the world. Hello guys. Some of them are doing their exchange semester here, and they often say that PBL helps them learn and retain things very easily. I can understand why. It's a very active way of learning because you have to bring your own perspective to real-life cases. You have a lot of freedom to manage your time, your studies, your hobbies, and your work. Of course, that also means a lot of responsibility. Right now, for example, I'm arranging my exchange semester in Madrid. How cool is that? In this group session, we're the managers who have to allocate the resources of a real company. This is how we put into practice what we learned this morning. Studying here means being proactive and learning to plan well. Prioritizing and performing well under stress are great skills that help you develop as a person. But now, it's time to grab a coffee at the Student Service Center.
Right now, we're at the Brightlands Camelot campus. Here we can apply knowledge from lectures and tutorials in a practical setting. This helps us understand what we have learned and further develop our lab skills. Today, we're determining the amounts of cholesterol in various products. What I really like is the project periods at the end of each semester, where we complete a full research project that includes planning, collecting data, analyzing, and presenting the findings. That way we learn how research works and we're able to see what it's like to be a real scientist. After practicals, I have to write a lab report that also helps me process everything I have learned today. UM has a lot of learning spaces where you can work on your own or with other students. This evening, I'm meeting my friends for a movie night organized by the MSP Study Association.
If you study law, you have to read quite a lot. Not all information is relevant, so you learn how to easily find the information you need to solve your case. In the afternoon, I have to give a presentation, so I like to practice it with a fellow student. Later today, a lawyer is giving a lecture. This will help us better understand the case we're working on. Speaking in front of a group is quite exciting the first time round, but you get to used to it quickly. And having to present helps you also to adopt knowledge better. What I like the most is that we sometimes get to enter a plea in front of a real judge. These moot courts are really exciting. Now it's time for drinks with friends. Studying is important, but so is relaxing once in a while.
Yes, I promise.
Dear Ms. Cao, here. The degree committee here present has assessed the quality of your thesis and in particular the way you have defended your thesis this morning. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you officially the degree of doctor. And I think you're pleased about this. Professor Heron is authorized to confer upon you this academic distinction in accordance with Dutch university customs, and therefore I invite your supervisor now to take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
Yes, I promise.
Then, by the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Jian-Hua Cao, the degree of doctor and grant you all the rights attached to by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary and the other members of the committee and affixed with the official seal of the university. Esteemed Dr. Cao, dear Jian-Hua, Gongxi, congratulations. You have done it. Today marks the completion of a 5-year journey that started in October 2017 after your first international flight away from China. You made a huge step moving from Beijing to Maastricht, literally a world of difference. You jumped literally into the deep end of the pool and started swimming. Already at the interviewing process for the CSC grant that you, as the outstanding student that you are, were awarded, you stood out. Contrary to some of the other candidates we interviewed, you were very communicative, and I think we've seen some of examples of that today as well. Maybe not yet perfect English, but clearly very knowledgeable and incredibly enthusiastic, and that has never changed, and I'm very happy with that. You were offered a position that started as a collaboration within the MEMC pathology group of Erik Biese to work on the development of 3D mass spectrometry imaging and the dynamics of arteriosclerotic plaques to visualize the complexity of plaque metabolism. And you've demonstrated that perfectly today. Eric unfortunately could not be here today, but I want to thank him and Peter Goossens and the entire team and his representative here for their collaboration that cemented the foundation of what you, Jian-Hua, have achieved today. From that start, your journey was a story of continuous and rapid growth. And how you have grown, you've demonstrated here once again today. You always complain that your English is not good enough. And that's a testimony how high you raised the bar for yourself. And I think today you've demonstrated that it's more than good enough. You push yourself to grow and tell me that I should not be so positive, as you think your achievements do not meet up with your own very high standards. You're continuously challenging yourself and the world around you, exactly what an academic scientist should do, and you do that really very well. And the good thing is, You have lots of fun while doing it. I think today again shows how much you've grown and into a really independent researcher and how engaged you can be in scientific discussion. Your personality has always been very energetic, and you have adapted very quickly to the Maastricht culture. From day one, you were outspoken and unusually direct. I absolutely enjoyed that, and it has helped us to reach several landmarks in your thesis more efficiently. It also helped us to grow as a team, together with Benjamin, your co-promoter, and our regular progress meetings very quickly turned into very lively discussions where your input was already very loudly heard. In the first, or maybe it was the second progress meeting, so this was in, I think, September or November 2017, you stated, I was a little bit confused about the pace of life here, and I became a little impatient. And I think that impatience never changed. You always wanted to move faster, quicker, and better. And I think you still do. So more than 3 years ago, you went back to China for Chinese New Year. And you brought a small gift to some of us, and I was lucky to receive a red Chinese knot. It's still in my room, at my home. This knot is believed to be a representation of the concept of balance, completeness, and good fortune. In your acknowledgements, you also point out some of the challenges you experience while moving to another country. I think such a move throws you off balance and might have an effect on your completeness, as you miss family and friends from before the move. But how you deal with such a big change also shows how incredibly resilient you are. And that is something you have demonstrated throughout your journey. Another testimony of your successful personal growth, and showing that you are, as Marie Curie said, really gifted for something that you've attained to today, Good fortune is also another essential element of success. And you had the good fortune to also work closely with many of our colleagues from the Department of Surgery, some of them present here today. And that have resulted in 2 chapters in the thesis: imaging fascial healing and studying stable isotope labeling for the metabolic dynamics of tumors. You very quickly adapted to these new themes, again showing your resilience and determination, and shows how much of a team player you are. And while your thesis is based on 4 publications, your publication list is actually extensively longer because you are such a team player and capable of respectively engaging with many different scientific teams. So that's about the science. But you're also a very social person, and especially interacted with what I call the French contingent. A number of French PhD students, some of them here today, some of them hopefully present online, who are still affectionately calling you Wawa. You traveled Europe with them and developed beautiful friendships with several members of the M4I team that I think will last you for life. You spent some time in the lab of one of our former colleagues, Martha Martin, even during the pandemic. And I want to thank Martha for her help in generating the data for the chapter in your thesis and her hospitality for hosting you there. And you do that as well, but I hope she's online. Thank you, Martha. I think one of the things what's sort of interesting being a Chinese student in the Netherlands, traveling to Spain in the middle of a pandemic, is getting vaccinated. And I think you must be the best vaccinated of the person of the entire group now, because I think you had 5 vaccinations already. I'm jealous. So you've already started as a postdoctoral fellow at M4I, working on the Leishman-Heiser project that you already are accelerating forward. I'm convinced that you will again take that project to successful completion of truly multimodal molecular understanding of this horrific disease that affects so many people in the poorest region of the world. Your engagement will definitely make that happen. Remember, you are already the scientist that did not only dream about something that can change the world, but you've actually done it here today. You have changed yourself. I want to end with expressing my gratitude for making M4I such a lively place with your personality and a fun place to work for all of us. I have truly enjoyed our journey together. Your spirit is contagious, and I hope that your spark will ignite many others. You have jumped into the deep and began this journey, not entirely sure if perhaps you could swim, but you've come out a champion at the end of the pool. You're awesome. So congratulations once again on this newly obtained honor. And I know that your family's not here today, but if they see this ceremony and your incredible success, I'm convinced they are as proud of you as I am today. You have a very bright career ahead of you, and I'm glad that it started and will continue for a little while in this beautiful city of Maastricht. And now, enjoy the party. Thank you very much. And I'll give the word back to the pro-rector.
Thank you very much. So, Dr. Cao, how does it feel?
I feel great.
Perfect. That's exactly what we would like to hear. I would like to congratulate you on behalf of Maastricht University, of our Board of Deans. And in my congratulation, I also bring forward your promotion team. They put all the effort in you for 4 years to bring you to this final stage. I also would like the members of the Corona who came today to ask for 6 minutes or 7 minutes questions, but it takes more time to prepare these questions than 6 minutes, I can guarantee you about this. I also would like to, those who are online in China, I would like to mention that we are feel very proud here that you came to us here as a CSC student. And I know there are quite some China scholarship students in the audience, and I would like to say this is the excellent example, and we're looking forward to your presentations here as well in the near future, and I'm looking forward to that as well. So after saying this, I think it's also time now to celebrate, and the celebration will be as follows. We are going as the members of the Corona here with the candidate to the stairs of our rector's here office, and we're going to make a picture, and you are already invited to enjoy before us some pie, some coffee, some tea, whatever there is in the rector's office. And saying this, I'm going to use my hammer. You haven't seen it, but I'm showing it to you here. And with this hammer, I officially close this ceremony. Thank you very much.
Thank you.
Grab your book. I want to drop this one. Congratulations with your phone. Congratulations, supervisors.